Welcome to Chris Khan Academy, the YouTube channel, which brings you about mathematics and biology. But now we are going to discuss about biology on the point called diversity of the microbial world. The diversity of microbial world. Subscribe, comment, share, and like. Follow us on our social media called Chris Khan Academy, so that we can catch up each and everything we post on our YouTube channel and even different social medias. Let us square one. Microbial world of the diversity of the microbial world. Here we are going to first start on the point called classifying microorganisms. So in the first chapter we discussed about different origin of the microorganism and what are microorganisms and what consists of microorganisms. So now we are going to see how we can classify the different microorganisms. So classification system arrange living organisms into groups based on common factors and show how they are related. So the way you classify the living organisms means that you give them to be in a certain community such that we can be classified there due to their different features that they met with the other organisms in the diversity. So arranging system is used to classify organisms according to their similarity. More similarity, lower the rank. So how they are similar, they lowers their rank. And taxonomy. So the point 3, taxonomy, the science of identifying and classifying organisms. This is the one system directly used by those scientists to classify different living organisms about what we call their order and families. They use what we call taxonomic hierarchy. So the classification of microorganisms generally has been based on the relationship inferred from observed similarities and different in morphology and physiology. Orderly, we focus on morphology and physiology of the microorganisms in order to classify them. What they do, their features and their shapes, how they react in the society they are. That gives the meaning or the way we can use to classify these microorganisms. So here we have taxonomic hierarchy constitutes of from the kingdom to the species. Here have kingdom, phylum, class, order, family, genus, and species. This is a hierarchy, taxonomic hierarchy. Even those microorganisms belongs in this order, family, class, genus, species, phylum, and kingdom. So here I have, for instance, an example of taxonomic hierarchy of Escherichia coli. The domain of that Escherichia coli is a bacteria, we know, and it's a phylum of Proteobacteria, belongs in class of Gamma Proteobacteria and order of Enterobacteria and family Enterobacteriaceae. Genus have Escherichia and the species name is what you call coli. These are different examples of microorganisms where they are likely to be one of the unicellular and multicellular, and you will see in there their genus name, their order, their class, and their species. This is an example of Paramecium caudatum. Another form of order, phylum, kingdom, phylum, class, order, family, genus, and species. We have Homo sapiens. These are human beings, how we evolved. We know as human being we belong in Animalia. Our phylum it is Chordata, and we have class of mammalians. So the order of primates and family Hominidae, and genus named is Homo, where species named is Sapiens. The scientific name is Homo sapiens. This is how we classify in due to the great taxonomic hierarchy. So here we have the second point in the classification of microorganisms, where you have classification system. The system likely to use while we are classifying the microorganisms. Two-kingdom system, plants and animal, based on the motility classification system, was known before the existence of microorganisms. We know that there is plants and animals. These are kingdoms we have at the first. So before the classification, we know these two. After the discovery of microorganisms, they were added into the classification system. As the third kingdom, we add up to plants and animal, then we add there what you call microorganisms when they discovered, and for simpler organization that lack tissues. So differentiation was recognized and called the protesia. So we have plants, animal. After checking up on the differentiations and we recognize that those microorganisms discovered have to be called what we call protista. Means that we have three: plantae, animalia, and protista. Protista, we could find there bacteria, fungi, algae, and protozoa. So this is the lack of specialized tissues. Let us see the way of modern classification of microorganism system. One significant differentiations become evident even among the protista. So also the first kingdom was proposed for the bacteria and called Monera. We find that different protista have different culture and ways of they survive. That's why they proposed to bring another kingdom where you can classify those Monera. During the period of 1959 to 1969, added a fifth kingdom of fungi. So the Winter's five kingdoms classification system quickly became the standard. This is our standard of five kingdoms: plantae, animalia, protista, monera, and fungi. So five kingdoms classification system proposed by Whittaker in 1969 and based on evolution of species from the prokaryotes, so along strategies for obtaining nutrition. These methods indicating that there were five major kingdoms: the monera, the fungi, the protista, the plantae, and the animalia. You see how they step one another from animal, plants, protista, monera, then we go to the fungi. So here we have five kingdom classification system. There are some ones we call prokaryotic, means they have one cell, other not having two cells, so cell nucleus. And here we have unicellular eukaryotes and multicellular eukaryotes. In prokaryotes we have bacteria, and in unicellular eukaryotes we have protista, where in multicellular eukaryotes we have plantae, fungi, and animalia. This is how we classify these kingdoms in three parts according to their cells. Modern as the three kingdoms classification system proposed by Woos in 1970s, based on the modern techniques of molecular biology, in particular the examination of the RNA, ribonucleic acid. Here we have three primary kingdom classification system. Here we start from universal ancestors, then we move to the eukaryotes. So before we forward to the eukaryotes, we have first Helicobacteria and Eubacteria. Those are bacteria. Here I found those photosynthetic bacteria and purple photosynthetic bacteria. We find that they have mitochondria and so on. So the phylogeny or evolutionary retardedness of the organism is often represented by a diagram of tree, and the trunk of the tree represents the main ancestor line, and the branches shows offshoots into specialized groups of organisms. The branches may also indicate origins, how closely related various organisms are. and an approximate time scale for the evolutionary history. This sort of arrangements place the more ancient groups at the bottom and the more recent ones at the top. So this tree diagram shows, starting from our ancestors until now, how the evolution was going to take place from the microorganism to the macroorganisms like plants and animals. Family tree of major groups of organisms. As you observe this structure, there is earliest cell. So after earliest cell, here we have initiated the bacteria, archaea and eukaryotes. So early eukaryotes, in the first step, the first cell appears in 4.5 billion years ago. The first eukaryote cells appears in 2 billion years ago. So how it is differentiated in two parts, the prokaryotes and eukaryotes. So in 2 billions and 4.5 billions, where you find those are in prokaryotes. Above of there, 0.6 billion years, the first multicellular organisms appeared as eukaryotes, and above that we have, starting from the protista, amoeba, animalia, fungi, and planta, which belongs in what we call eukaryotic cells. So let us focus on the survey of microorganism. A cellular microorganism require interaction with other organisms. This is what we call survey of microorganisms. This is called host to survive. So they are harbored and nourished by their host. Microorganisms depends on the host they are in. They don't make their own foods, and then they don't have complete what we call complete cell organelles that allows them to accomplish a different metabolisms of the life to get each and everything. That's why they depend on the host cells. When these cells, cellular microorganisms, are replicated within the cell of the host organisms, so they use the living cells' metabolic functions. Often in doing so, they disrupt normal cellular function, producing disease in those organisms. So as you see, they have to change the metabolic system of the organisms that are being hosted in, and then invade in the nuclear materials, those DNA. Then they challenge there. Then they have to start to control the cellular metabolic functions. That's why they disrupt normal cellular function and producing diseases in those organisms. So as there is what we call cellular microorganisms, as the point we are. In cellular microorganisms, you find there viruses, viroids, virusoids, and prions. So there are different characteristics that present this. Obligate intracellular parasites, high specificity, and there are difference between these in structure and host cells. So here, starting from the virus, including animal virus, plant virus, and bacteriophages. So this is what we call virus. Then if we say the viroid, a prion is a type of a protein that can cause disease in animals and humans by triggering normal healthy proteins in the brain or to the fold abnormally. So the prion mode of action is very different to bacteria and the various viruses, as they are simply proteins devoid of any genetic materials. So here in viroids cause more than 16 different plant diseases, where I find that those viruses are likely to include animal virus and plant viruses, even bacteriophages. If we say the virusoid, they are pathogenic RNA that can infect commercially important agricultural crops and are the virusoid, which are subviral particles. So the presence of the herp virus and infect host cells in the existence of the herp virus. Then if prions are discovered by Stanley B. Procina and Department of Neurology, and was in University of California, the School of Medicine, San Francisco in USA. So it was in 1997 Nobel laureate in medicine for his discovery of prions, a new biological principle of infection. So prions cause scabies, mad cows disease, also bovine spongiform and encephalophytes, and so on. The different diseases caused by these prions. At the time, scientists believed that any infection particle must contain DNA or RNA. Then in 1982, Stanley Procina, a medical doctor studying scabies, discovered that the disease was caused by proteinous infectious particles or prions. Proteinous infectious particles, because proteins are acellular and do not contain DNA or RNA. So Procina's findings were originally met with resistance and skepticism. However, this research was eventually validated, and he received the Nobel Prize in Physiology or Medicine in 1997. So this was about what we call prions. There is this consists only of short strand of the circular RNA capable of self-replication, and the first video discovered was found on the cause potato tubers, spindle disease, which cause slowly splitting and various deformities in potatoes. The structure of virus. So